Hi everybody, this is Dr. A and we're going to talk about the role of microscopy in medical microbe. So as a reminder, for laboratory diagnosis, we utilize direct examination. So in micro would be looking at the microscopic morphology, so the colonies on the different plates and reporting that. And then the microscopic morphology using various stains. And this is a fungal stain, for example, of lactophenol cotton blue, and looking at the slides under the microscope, which is going to be the focus of this lesson. We often grow bacteria that can be grown, and some of the yeast that can be grown, so that we can further identify them. And we can identify them by biochemical reactions, but also via nucleic acid detection. Think some of the rapid tests like the BioFire, for example. The identification that correlates then of the direct examination, so microscopic and microscopic, their growth characteristics on the agar, maybe if they exhibit hemolysis on the blood agar, or if they turn a certain color on a differential media. And the biochemical reactions, so these are represented here. There are different formats and different brands, but the idea is the bacteria can be identified by the sugars they ferment or the amino acids they use, and these can be used to confirm the identification from nucleic acid techniques like the BioFire. But then we can also get antibiotic susceptibility in some of these panels if they're set up, obviously. We can also characterize them by their antimicrobial, antifungal, or antiviral susceptibility to know what the best treatment will be for the patient, but it can also give you clues as to the identity of the microorganism. So microscopy is the use of a microscope to magnify objects that are too small to be visualized with the naked eye. It can directly detect microorganisms in clinical specimens. So think of a swab, let's say a vaginal swab, and you do a gram stain on it, and you see some gram-negative diplococci inside of white cells. Well, that's a direct detection of microorganisms that is suggestive of gonorrhea. And so you can see if there are multiple morphotypes or if there's a single most prominent potential pathogen. So you can see if it's maybe a mixed flora, and maybe it's just a contamination of skin flora, or maybe you see specifically a lot of gram-positive cocci, for example. Again, you can also do a preliminary detection of organisms that are not easily cultivated, again, such in the case of gonorrhea. And some of the parasites also, for example, if you saw trichomonas, that would be, easily identified on a microscope slide. We don't cultivate those guys. And you can also evaluate specimen quality to make sure that the swab was appropriately collected, or the specimen was appropriately collected. And that, for example, if it was a pussy specimen, that there's white cells and bacteria and stuff in there. And then you can also use microscopy to characterize organisms that are growing in a culture. So we can get some of the colony or some of the blood from a blood culture, and we can make a slide, and then we can see what organism is growing. It should usually be a single one, but maybe there are several on a plate, and we can then see what each one are. And we can see, correlate with the way the colony looks and its growth characteristics, and we can see if it meets what we expect it to be, on the gram-positive, gram-negative reactions and its morphotype. So that is called an indirect specimen, or an indirect microscopic evaluation, and it's done again from the growth plate from the blood culture or whatever you've got growing. Bright field microscopy is what we use the most. It is what you think of really when you think of a microscope. We use visible light, It passes through the specimen that is usually mounted on the slide and through a lens that's in the objective, and it is magnified and therefore visible to the naked eye. There are three main principles to microscopy: the magnification. So the objective lenses used, for example, are listed here: 10x, 20x, 40x, and 100x. I've seen 4x's also in our teaching labs. We have them. At one of the hospitals that I work, we had a 50x oil immersion, so 50 and 100 were oil immersions. Again, these vary with the microscopes and your needs and within your bench or hospital. The ocular lens is always 10 times magnified, so that's the lens in which you look. And then your total magnification is going to be then the magnification of the objective lens multiplied by the magnification of the ocular lens. So if you are using the 10x, it would be 100 times multiplied. If you're using the 4x, it would be 400 times multiplied. If you're using the 100x, it would be 1,000 times multiplied. The resolution is the detail of the magnified object. And so it has to do with the closest distance between two objects that allows you to see each object as a separate object. The resolving power is a characteristic of the microscope that's based on the manufacturer, and they are the ones who determine it. It often has to do also with the cost of the microscope. Immersion oil is used for the oil immersion 50x and 100x to increase the resolution, and so you get a sharper image of the tiny microorganisms that you're trying to look at. Contrast in a microscopy can allow objects to stand out from the background. We tend to try to increase contrast in several ways. So you can use stains to contrast objects from the background, and then you can also, in the wet prep, drop the condenser some and close the diaphragm some, and that can increase the contrast where you can have a darker, like a ring or something around, or you can, the darker edge of the cell is darker so you can see it a little bit better. You increase the contrast. So a typical bright field microscope will have a light source at the bottom that shines up through a condenser lens that has an aperture diaphragm that can be open or closed. You can increase or decrease the intensity of the light there, but you can also increase or decrease the light intensity, usually with an adjustment knob. And then the condenser shines the light and focuses it here on the microscope slide. The specimen is mounted on the slide, which is loaded on a stage. There's a mechanical stage that can move the slide back and forth. And then you can select an objective lens, and if you have an oil immersion, you would put oil on the slide, and then you will look through the ocular lens at the specimen. And so the images are flipped as you look at them, so you have to remember when you use a microscope that right is left, left is right, up is down, and down is up, kind of like Alice in Wonderland. Kohler illumination is a method to provide the maximum illumination and resolution for your microscope. It properly positions the condenser to ensure that light is focused through the specimen. So this would be an improperly aligned microscope, and so the way you do this is you load a slide on the microscope and you focus on that slide. It's ideally you use it, I do it at 4 or 10x. 10x is better. So you get focus on the slide, and then you close the diaphragm, and you should see the circle or the hexagon of the diaphragm. And if it's out of alignment, it's going to be fuzzy, like here, right? And so you are going to mess with the little screws that are at the condenser, and they can move this. So you have to go gently, and you're going to move the field to the middle and make it look like this right here. So you have to make sure that you see these sharply, and there they are. And then once it's centered, you can open it back up, and then you should have a clearer, crisper image. So again, direct and indirect smears, we've already just kind of hit up at this concept just a few minutes ago. If it is from a primary clinical specimen or sample, it is a direct smear. It identifies the types of cells present in the sample. So in there, you're looking for the presence of white cells, or some yeast, some fungi, bacteria. Like, what are you seeing? You probably can see some epithelial cells, maybe some mucus and stuff. And it's used to provide preliminary information to begin patient care and to determine identification methods. So if on the direct specimens of a wound we see some gram-positive cocci, we can report that. So we would report preliminary, on the preliminary report, gram-positive cocci in maybe pus cells or white cells seen on the gram stain of the wound smear. And so then you need to set up a gram-positive ID panel for your biochemical ID, and your physician knows that he can at least select some antibiotics that would target gram-positive organisms. And then we set up the culture on the plates, and if there's that organism, the gram-positive cocci, grows, and we can see if it's all the same colony types, they all look identical, then it's the same morphotype that turned into the growth on the plate. And then you can gram stain specifically that colony to verify that you still have those gram-positive cocci. If it was a mixed culture, because sometimes it can have infections with multiple organisms, you could differentiate them on the plate and then gram stain each of the types of colonies that you see. And so that's an indirect smear because it is directly from a colony. And then you can use this to correlate with the initial preliminary direct smears and resolve any kind of discrepancy if you note any. The gram stain is the primary staining method that's used in microbiology. It is a differential stain used for the examination of bacteria, although we can detect yeast and parasites and other things with it too, of course, if they're in the specimen. The clinical material is fixed to a slide before staining, so that could be the swab, like the wound swab, it can be rolled on the slide, and then it can be heat fixed or methanol fixed or whatever your hospital uses for their protocol. And if it's off of a colony, then you pick a little bit off of a colony, put it on the slide, mix it with a drop of sterile water, get a good spread out, and then you let it dry and you heat fix it or methanol fix it. Then your primary stain is a basic dye called crystal violet, turns everything purple. The mordaunt, which makes the gram stain stick, so the crystal violet stick is called Gram's iodine. And then you have to do a decolorizer to remove the stain from the gram-negative organisms. We usually use alcohol or acetone, and it's often in a 1-to-1 solution. You want to drip it slowly at a 45-degree angles for maybe 20 to 30 seconds. You want the decolorizer to run clear. There's also wash steps in between each of these stains and the decolorizer. And then the counter stain is a basic dye called safranin, and that is to be able to give color to everything that lost its color during the decolorizing steps, so we can visualize it. And so in the gram stain, safranin is a pinkish red color, and so everything that stains with the crystal violet is considered gram positive and it's purple, and everything that stains with the safranin is a pinkish red color and it is considered gram negative. And so here's an example of gram positive rods on this side here, nice pretty purple color. Sometimes if the stain's really deep, it could even appear like they're almost black, but this is purple. This is pink or reddish pink, and this is a gram negative rod. So some of the limitations of the gram stain, if the smear is not prepared properly, so too thick or too thin, sometimes it can be difficult with things like spinal fluids if they don't have a lot in them. Ideally, you want to concentrate everything on there. if it's too thick, or if you grab too much of a colony. Sometimes I have students get a little too happy with that and just grab like too much bacteria. And then even with the water, there's not enough to thin it out, and then you can't really see the individual bacteria because it's so thick on there. You could over or under decolorize, so it could lose too much of the purple or not enough of the purple. Sometimes the dead or dying cells can be hard to interpret. The heat fixation can distort cells, especially if you go too fast. So sometimes patience is a virtue to get a good stain. Sometimes there can be some artifacts in a gram stain, maybe some precipitates and stuff from the stain that just junk up the slide. And then, of course, it's really only as good as the quality of specimen that you have. The acid-fast stain is probably the next most used stain in micro. It's a differential stain that's used to detect bacteria whose cell walls contain mycolic acids. So that means for us, Mycobacterium, and most usually used for Mycobacterium tuberculosis. There are two methods: the Ziehl-Neelsen stain. The primary stain has to enter the cell wall by heating, so there's a heating step that's associated with the primary stain. In the Quinone stain, the primary stain contains phenol, and that's a cold method, so you don't have to heat it. That's the one that we use in our lab. The results: the acid-fast organisms will stain pink due to the carbofuscin, and the other objects will stain the color of the counter stain, which could be blue if you use methylene blue, or green if you use malachite green. This is a typical acid-fast. This would be a positive specimen with the pink carbofuscin. This would be a negative specimen that appears blue as the counter stain. So phase contrast microscopy can be used also, especially useful for wet mounts, and you can visualize movements. So the beams of light pass through a specimen to create different light intensities and greater contrast. You do not need staining, therefore living organisms can be observed. Some microscopes can use some of the phase contrast for crystal analysis in body fluids, but for us in microbiology, it's mostly to visualize organisms that are alive. Fluorescent microscopy. So this is being used increasingly. The fluorochromes are raised to higher energy levels after excitations, and so these are components that fluoresce, molecules that fluoresce. So what happens is you basically bombard the fluorochrome with high intensity light, and then the excess energy is released in the form of visible light when the molecules return to a normal state. The best way to explain fluorescence to me is if you've ever played with glow-in-the-dark little stars or any kind of glow-in-the-dark thing where you have to shine a flashlight or leave it in the sunlight to charge it, and then if you put it in your room or whatever, you turn all the lights off and make sure everything's dark, and you can see it glowing and emitting light. But eventually, of course, it emits all the lights, and then it becomes back to normal. And you have to recharge it again. And so we can do that with microscopy. We can hit the light indirectly in there and then detect the light that's emitted from the fluorochromes. The objects will appear brightly lit against the dark backgrounds, and you can use different type of fluorescent molecules with different colors like greens and reds and yellows and orange. And so here again is the way it would work with the microscopy here is that you would have a light source in the excitation light, which is reflected off of a light wave splitting mirror and charges the specimen. So the yellow light is a light that charges it up, and then the fluorochromes then will emit the fluorescent light, which is represented by the green beam, through a barrier filter, and that's what you would visualize. And so there's two ways this can work with the microscopes. Is you can put a fluorescent dye on the specimen, and it will bind to the target bacteria or yeast or whatever, and it will all glow regarding of the morphotype. So it's just if it's there and close, so you can visualize it in tissue specimen or whatever. And or you can do immunofluorescence where you there's a fluorescent dye that's attached to an antibody, and you incubate your specimen with the antibody, and the antibody was specifically attached only to the antigens for which it was made, so it will only identify a specific type of bacteria that you're looking for, and it would bind to it and glow, and you would only see glow if that specific bacteria that you're looking for is present in the specimen. The fluorochromes are acridine orange stain. It stains nucleic acid. It is a non-specific cationic dye. It confirms the presence of bacteria in blood cultures when blood cultures are indeterminate. So it just says, hey, there's some bacteria in there. Chalcofluoral white, it binds strongly to cellulose and chitin, meaning it is used to identify yeast and fungi. It greatly enhances fungal visibility in tissue and other specimens, makes it glow really, really bright. And then auramine, rhodamine stain is a basic dye that binds to mycolic acids, and the cells will appear bright yellow or orange against a green background. Dark field microscopy is directing light at an oblique angle to achieve contrast. The only light that hits the object will be visualized in a dark background. And so basically in light microscopy, the background is light. In dark field microscopy, the background is dark. And the edges of the and features of the organism you're trying to visualize are what's catching the light. And so like if you think of with a light field, to increase contrast, you can, close a little bit the diaphragm and drop the condenser, and that'll make the edges of the cells dark. Well, this one is flipped. With dark field microscopy, the edges of the cells will become light. And it's very similar to bright field or light microscopy. There, all the light comes through the specimen, through the objective, etc. Whereas in dark field, you have a filter here, something that blocks the light in the middle, and it's only the light at the edge that angles and crosses and hits the specimen. Again, the direction of the light beams, you can see there's a dark field ring in the middle of the condenser lens, and it hits, which means the lights that come through the specimen hit at an increased angle, and that's what makes the edges of the cells and the components you're trying to visualize visible. Here's an example again of a dark field microscopy slide. And then lastly, we're going to talk a little bit about digital or virtual microscopy. It is automation and digital imaging combined, and so you can combine that with some automated staining also. And then the microscopes, or however it's built into the system, will take pictures of all the fields and recorded fields, and it's really nice because then you have a digital record of the images in the microscope, and you have the ability to adapt these digital microscopes for multiple uses. You can use it for bright field, phase contrast, and fluorescence. It can be used for slide scanning and to coordinate tracking of specimens throughout the system, especially with an automated, more of the automated systems, which are the large expensive system like the BD-Kiestra in microbiology. It can standardize a technique, and you can run quality control and stuff on that, but it helps control the better quality of a specimen if it's the staining is automated, the microscopy is automated. You don't have variation between the techs and how well they're trained and what their background is and what they've learned, etc. It also allows for consultation and transmission in real time of images and results, so that you could send them to a pathologist, to an infectious disease doctor or something for them to look at. So this is done in a lot of other realms, to just a digital imaging and consultation, especially our friends in radiology have been doing this for a long time. And there are some of these also doing in hematology. We're taking pictures of cells for records, etc. And so we're really expanding the use of the digital microscope for better record keeping and tracking and history of the specimen, etc. So, all right, so that wraps up our lesson on microscopy, and I hope that was helpful for you.